All right. Well, we have Dr. James Bianco from TuHURA Biosciences with us. And last year, his company signed a deal to acquire Kineta's Anti-VISTA antibody. Now, VISTA is a checkpoint inhibitor that's probably not as well known as the PD-1s and the PD-L1s like Keytruda or Tecentriq, but it is compelling. And Jim, why don't you tell us why you were so attracted to this anti-VISTA monoclonal antibody that you're supposed to pick up pretty soon and add to your pipeline?
VISTA is the only checkpoint that's expressed on what are called quiescent cells, meaning that cells that aren't actively dividing. All the other checkpoints that you mentioned are present on T cells, activated T cells. And so VISTA is a very unique target in that it is concentrated in a compartment, i.e., the bone marrow cells, that typically is where you see conditions like leukemia. And in particular, while Kineta really focused on targeting solid tumors, our focus was on some emerging research that shows that VISTA is highly expressed on leukemic blasts. These are the leukemic cells, particularly in a type of leukemia, the most common called AML, that's seen in adults. And it's believed to be why immunotherapy doesn't work in leukemia. In fact, all the checkpoint inhibitors you mentioned are not expressed in leukemia, and therefore having a checkpoint inhibitor against them won't work. Whereas VISTA now is believed to be the primary reason that leukemia escapes immune recognition and essentially either results in poor responses to current therapy, and certainly for why that leukemia persists, comes back, and ultimately results in the death of patients, in the majority of patients with AML. So we think it's a very unique target. The research that's coming out of a number of centers are showing as cells mutate to become leukemic, this particular target starts to appear more and more on those leukemia cells. And so if you're able to block it, it may work very synergistically with current therapies or even new therapies that are called menin inhibitors. A lot of interest in the hematologic community about these particular drugs because they're associated with certain mutations in genes that drive leukemia. And what they've recently showed at the last hematologic meetings was that those mutations not only drive leukemia, but they drive VISTA expression on the leukemia. So now you have a genetic marker that's associated with a target that we will have control of with the VISTA inhibiting antibody. So that's a very exciting potential therapeutic advance to combine VISTA with this new class of drugs called menin inhibitors to see if we can get better response rates, but more importantly, long-lasting response rates, which is kind of the Achilles heel in leukemia. It's why they relapse, and ultimately there are no subsequent therapies for them.
Right now, you talked about combination therapies, and I think there's been some early work done with the previous owner of the asset, along with Keytruda, actually, with their anti-VISTA asset. And also it was done in solid tumors. How does that work help what you're doing at TuHURA?
I think twofold. The one thing they showed us was that you can give this antibody at very high doses. They went to 1,000 milligrams given every 2 weeks. That's a high dose of an antibody, but importantly, unlike the first generation of VISTA antibodies, this was extremely well tolerated, didn't see severe side effects associated with it. And if you looked at blood levels of the antibody, they were able to show that they shut off the VISTA target 100% over the entire 2 week period of time. So that data is important for the development into the next phase of study with it. They've also demonstrated that they can give it safely in combination with important classes of drugs like Keytruda, like a PD-1 inhibitor. And so if you start to look at future studies in what we call solid tumors, as opposed to leukemias, which are termed liquid tumors, there are certain tumors, in particular endometrial cancer, which expresses a high level of VISTA, again, as a way for that tumor to escape immune attack. But it's also expressing high levels of the target for Keytruda. And so Keytruda works very well in the frontline treatment of endometrial cancer, but as they progress and relapse, VISTA is the primary culprit that creates resistance against Keytruda. And so that's a logical, scientific-based combination to look at patients who progress on Keytruda with endometrial cancer for giving the anti-VISTA antibody alongside of the anti-PD-1 antibody, i.e. Keytruda, kind of again like a 1-2 punch, trying to prevent the tumor from developing these protective mechanisms to escape immune attack. So now you have two ways to block their ability to do that.
I believe you're going to have a small proof of concept trial that's going to help you decide what the next direction that, where you go. What is the plan for that? And if you and as you said, I think if it's successful, then you're going to be able to get expedited treatment for that too, similar to maybe what you're going to get with IFX 2.0. Is that a fair way to look at it?
Uh, yes, John. So I mentioned this NPM1 mutation,
Hmm.
right? And this was a discovery that led to the development. I've mentioned these menin inhibitors. Why menin? Menin is the final common pathway that generates the signal that makes those cells leukemogenic, i.e., malignant. And so if you blocked menin, they were able to show about 25-30% of those patients actually would get a remission. And while that is important, unfortunately, those remissions didn't last very long and those patients relapse. But it was a major advance because normally chemotherapy wouldn't work in those patients. In fact, they just failed chemotherapy. Well, the data that I was referring to that came out of a number of institutions say when NPM1 is mutated, it not only produces menin, but it produces VISTA on the leukemia cells. And so if we can combine VISTA with menin and we compare it to the results for patients receiving menin alone, because menin now will be the standard of care in patients with that mutation in first failure for AML, first failure treatment for AML, and we can increase that response rate from 30 to 40 or 50%, that's a meaningful improvement in a disease that subsequently, when they relapse, hope no alternative therapies. And that's typically that unmet medical need population that the FDA is so keen on helping sponsors develop drugs that may address that need. And that is the purpose of their accelerated approval pathway. So that trial won't require a lot of patients, right? Maybe 20, maybe 30 patients where half get menin, half get menin and the VISTA inhibiting antibody. The readout's very quick. These patients are either going to go into remission quickly or they're not, quickly meaning 30 days, 60 days, 90 at the most. And that will give us the ability to take that data next year. So we'll start that trial, we think, in the third quarter, late in the third quarter. Uh, this is, again, everybody is using these drugs in that patient population, so it won't be hard, essentially, to find centers that want to participate in the trial. So having data by mid next year, we can be in front of the FDA in the second half of 2026.
All right. Well, great. Well, we had Dr. Jim Bianco, the CEO of TuHURA Biosciences, with us, and ticker symbol HURA.